I'm going to actually share my journey out of Africa and how I got into a career in life sciences. So I was born, as you heard, in a small town beside Lake Victoria. It is south of the equator, about 18 miles south of the equator. And this is a picture of me with my father outside what we call our tin house. This is a one-bedroom house where I was born. And the thing about growing up in this place called Kisumu was it's beside the second largest lake. It's Lake Victoria, and there are a number of interesting living creatures in this lake. One, there are hippos, and the thing that I found exciting was that there are this fish, cichlid fish. They're very fast evolving, and there are about 500 species of this fish in this lake. This is now one of the really key models for understanding how evolution works. And there are some people in Cambridge who are studying this. I went to Kisumu High School, and that's where I was lucky to have a biology teacher who inspired me to take interest in living things. So that was an important part of my growing up in this very small place. But I actually didn't meet anybody who had been to a university. My parents hadn't been to a university, so I didn't have any role models. I didn't understand how you could get into a career in life sciences. Now, at that time, in 1963, Kenya was approaching independence. So there was quite a lot of uncertainties about what you do. The situation was difficult. So my parents sent me to England on a one-way ticket. And my father told me to go and get some education because that's how I might survive. So I went there without a plan and an uncertain future. I went to 2 universities, acquired some education in biochemistry and natural sciences. And then the situation was getting a little bit difficult in the UK because there were a lot of immigrants coming from former colonies that were becoming independent. So I actually decided to leave UK. I went to Paris for a couple of years. And while I was there, I met somebody who became very important to me in my career. And so I ended up in Cambridge, actually, in 1972, to do a PhD. So the person I met is Bob Edwards. He's the pioneer of IVF, in vitro fertilization, IVF babies. So he was, at that time, when I went to Cambridge, he was working on IVF. My project there was to study the mechanism of how the embryo implants, blastocyst implantation. This was actually a big problem for IVF at the time. So I was working on this problem in rats to understand the mechanism of how this embryo can implant. Actually, I didn't get so excited about IVF, but at that time there was some pioneering research that was ongoing to do with how mammals developed. This was a new field at that time. So I became fascinated with mammalian development. And in fact, Louise Brown, the first test tube baby, was born in 1978 anyway. So I thought the problem was, in principle, was solved. I wanted to do something different. So I'd actually started working with a friend called Matt Kaufman. And what he got me excited about working on these embryos, parthenogenetic embryos, which are unfertilized. And so we kind of became interested to think about whether mammals can develop asexually without fertilization of an egg. And Because the idea was that, there are many vertebrates like lizards and frogs that can develop without fertilization, passed genetically and sexually. And actually what happened at that time, there's a very strong debate about things like the redundant male. There was a strong feminist movement at that time in Cambridge. So there was this idea that, the males were redundant. You could develop human beings without fertilizing eggs. So this was the kind of the background to all of this. So I actually formulated this question in my head that, could mammalian embryos develop without fertilization, and whether you needed a paternal genome? It's a very esoteric idea, but this is something I got absolutely obsessed with. So fortunately, I got my first lab. I started in 1979, just outside Cambridge, and I got this position, which also was extremely fortunate. Because when I got to this institute and started my first group, I could do whatever I liked. I was not restricted from what subject I could follow. So I went to Animal Research Station with this question in mind about trying to explore this question of asexual reproduction in mammals. And to cut a long story short, after a lot of experiments, most of them failed, we came to the conclusion that you cannot reproduce mammals asexually. But what we became aware of was that if you make these embryos, which are genetically identical, genetically similar, with two maternal genomes or two paternal genomes, they will not develop. They stop developing after a certain time. So you needed a maternal and a paternal genome for normal development. Now this may sound quite obvious to you, but according to Mendelian laws of genetics, all three of them should develop because according to Mendel, the genomes are equivalent if they're genetically similar. But we only found that the embryos that could develop would have a male and a female. So this was the precursor to the discovery of genomic imprinting. And so what became clear in around 1984 was that the parental genomes in mammals are, although they are genetically similar, they're functionally different. They function differently. And so what we found was that the genome that comes from the father is much more important for the development of the placenta. And the genome which comes from the mother is much more important for the development of the embryo. And so that's why you need both of them. So this was a really quite amazing kind of idea that this experiment told us. We had no explanation for this because we couldn't understand why this should be, because this was going against the laws of genetics established by Mendel in the 19th century. So although we were excited to see this kind of result, and we thought, this was something quite interesting and something new, we had no explanation for it. And when I first started giving talks, I had a lot of hard time from people because they just didn't believe it. So we needed to actually look and find an explanation for this observation. I'm still trying to find the explanation for this observation. There are still many things that are not known. But let me actually try and guide you through some critical things. This is a slightly complicated slide, but what we found was that although the parental genomes are genetically similar, they're epigenetically not similar. They are asymmetric. So the maternal and the paternal genomes have epigenetic asymmetry, and that's why you need both of them. So if you look at this chromosome 7, just taking one example, there are certain genes on chromosome 7 which are only show expression of genes that are coming from the father. Here are the examples. And this gene, IGF2, is expressed only, the copy that comes from your father is expressed, the maternal copy is silent. if you look at the maternal chromosome 7, there are different genes that are expressed similarly. And so this is where you get the asymmetry. You're having some genes that are only expressed when they come from the mother and others that are expressed only when they come from the father. It's called monoallelic expression. So if you look at this region here, we get an idea about the molecular mechanism of how the imprinting might work. I'll just take you through this. This is an example. There are variations on this thing. So there are these two genes, IGF2 and H19 on this chromosome 7. And this region here is called the imprinting control region on chromosome 7. And if you look at this region here, what you see is there are these marks. These turned out to be DNA methylation marks of CpG dinucleotides. So this is the imprint signature that goes in this case, on the chromosome that comes from the father. So this is the imprint that comes in from the germline, and then it's inherited. It's heritable. It comes in the egg, and it's heritable throughout life. So if you take your skin cells, you will see this imprint that has come from your parents' germline. And what this imprint here on this chromosome 7 is doing is that it allows these regulatory elements called enhancers to activate this gene H19, which is on the maternal chromosome. But the same enhancers will activate IGF2 on the paternal chromosome. So you can see that on chromosome 7, you have two genes. One is expressed when it comes from the father, and the other is expressed on the chromosome that comes from the mother. So this is the principle which applies to other regions, other chromosome regions, which work in similar ways. But there are variations on this. But so this is just to give you an idea of how this imprinting works. So if you now go forward, what we now know is that there are approximately 200 imprinted genes in mammals. So this is 200 out of 20,000 genes, which is about 1% of the genome. So it's a small fraction of the genome which is imprinted in this way. And there are these 200 genes. And they have a variety of functions, from regulating development, growth, physiology, brain development, behavior, and so on. So they don't have specific functions, they have a variety of functions. So this is how imprinting works. There are genes that come from mother and father, they're working differently, but they're important in terms of balancing growth and other aspects of mammalian physiology and development. So I'll give you just two examples of what these functions are. And this is one of the examples. This is a gene called PEG10, which comes in and is active when it comes from the father. The maternal copy is silent. So if you mutate this gene, what you see is that the placenta doesn't form very well, and these embryos don't develop fully, and they stop developing, because when you mutate this gene, the placenta doesn't form. So you can see that this gives you an idea of how this differential expression of imprinted genes can impact on development. The second example are these genes, two genes called PEG1 and PEG3. And this shows the expression of this gene. It's expressed in developing brain. These experiments were done in mice. So in mouse brain, you can see expression of these two genes. And what was found is that if you mutate this gene PEG1, it affects behavior, maternal behavior. So what happens is that when the pups are born, the mother doesn't show a normal maternal response, doesn't build a nest. So these pups are left, and they would die if you don't rescue them. So this gives you an idea that this is another gene which is having a completely different function. It's affecting brain development and behavior. So these are just the two examples of the 200 different genes having different functions. So this gives you a broad view of how imprinting works. I should stress one thing to remember is that these imprints do not alter the genetic sequence. They are marks that are put on without alteration in the genetic blueprint of the genome, so that stays intact. These imprints or imprinted genes, this kind of signature of imprinting, is heritable from eggs to adulthood. They persist. And there are specific mechanisms that recognize these. These genes called ZFP57 and ZFP446 are two genes which will recognize these imprints wherever they are. And this information is then transmitted from the egg to adulthood. And they will perform a variety of functions at different stages of development. So, coming from a very embryological experiments and observations, we now have some understanding of the molecular basis of how this imprinting works. And in 1992, I moved back to the university where I was appointed as Marshall Walton professor. And so I started doing some teaching. I was teaching natural science and medical students. And again, I was fortunate to have my own lab. So I started to expand my research on imprinting and on epigenetics, which was the new field now, which was developing, coming out from imprinting work, and also working on the germline and early human development. So we are expanding our research and going into different areas. And one of the areas that we started to look at was the germline, which is a lineage which gives rise to sperm and eggs. This is the germ cell lineage. And as I told you, we have, when the egg is fertilized, we have maternal and paternal genomes, which is the same in humans, and they also have functional differences and inheritance of epigenetic asymmetry. Now, all of this epigenetic asymmetry is programmed during the time when the germ cells are developing. So it's the same in mice and humans, but this is an example in humans. So the primordial germ cells, the precursors of sperm and eggs, in humans form during the second week of development, second week of gestation. So it's very early that the first cells appear, which is going to give rise to the next generation and the generation after that. So these are very important cells, set aside very early in development. And as these cells start to migrate towards the developing gonads, the first thing that happens is they start erasing, all the imprints are erased so that everything is wiped out by the time they get into the developing gonads. And when you start to get formation of sperm and eggs, new imprints are put on, depending on whether the embryo is a male and forming sperm or the egg. And then when fertilization occurs, you recreate this asymmetry. So this is something that is repeated for each generation. It's not something that's heritable. It is wiped out and reintroduced. And the same thing that I showed you in mice works in humans. So in humans as well, you have the two parental genomes which balance development of the embryo and the placenta, and you need both of them for normal development of humans. If by chance you have fertilization of an egg without a maternal genome there, and you have two paternal genomes, what you see is excessive proliferation of the placental tissue, and you don't get any fetal tissue. And conversely, if you have two maternal genomes, you also don't get development because there's no placenta, so you can't implant. But sometimes these embryos stay in the ovary. They form what I call ovarian teratomas and just contain fetal tissue. So what I've told you applies to mice and humans and all the mammals that have been looked at so far. So this is something which is universal to all mammalian developing systems. And as in mice, these imprinting has diverse functions in humans as well. So for example, it's involved in regulating development and growth, regulation of placenta and fetus, proliferation and differentiation of cells as they develop. It's also involved in energy homeostasis, brain and behavior, and endocrine functions. So there's a very diverse range of functions that's performed by this 200-odd genes. So they are very important. Now, if imprinting breaks down for some reason and you get disruption of imprinting, you have human diseases that form. So here's one example of what happens if there are imprinting errors on chromosome 15 of humans involving this gene called SNRPN. And what happens is you get this syndrome called Prader-Willi syndrome, where you have delayed puberty, obesity, type 2 diabetes, behavioral issues. So you can see that imprinting is important for development, but if something goes wrong, there's a cost. You have human diseases, and it's estimated that there are about 14 diseases that are caused when you have errors in imprinting. So the one sort of question which is really quite fascinating and need to find answers to is on the evolution of imprinting. When did it evolve, and what does it tell us about mammalian life and mammalian development? So it's supposed to have started around 160 million years ago. This is before the split between marsupials and what are called eutherian mammals. So this is the rest of the mammals versus the marsupials. This is 160 million years ago in the Therian lineage, when the imprinting was thought to have been initiated. And that is split into two, marsupials and eutherian mammals. And so what this tells us is that imprinting somehow is associated with what is called placental viviparity, which means that there's a development of embryos in utero resulting in live birth. And so it's thought that imprinting, through its influence on placental development, was important for this placental viviparity development in utero. And so there was, 60 million years ago, there was mass extinction and the dinosaurs were wiped out. And then this gave opportunity for mammals to proliferate and diversify. This is called adaptive radiation. And after this time, over 66 million years, we have huge diversity of mammalian life form, which you see now in the world. That is, to some extent, also happened with the marsupials, not to the same extent. Now, there is another group of mammals, which are called monotremes, which are creatures like echidnas. So these are egg-laying mammals, and so they don't have imprinting. So they're split off earlier. So what we can do now with the information that we're starting to collect on different aspects of imprinting, we can start to sort of piece together how this imprinting evolved and maybe understand better its significance, importance, and so this is something that I think might be interesting to focus on for the future. So I think imprinting, which started as a very esoteric idea, very curiosity-driven research, I believe has had quite a big impact, and I just want to illustrate different aspects that have been influenced by this discovery. So, as I told you at the start, this phenomenon, this functional differences between parental genomes, is due to not genetic changes, but due to epigenetic changes. So these are modifications of DNA without alteration of the genetic code. So this actually, discovery of genomic imprinting is very important for this whole field of epigenetics, which is now an extremely important area of research, and both in terms of understanding development and disease. So this is inheritance without changes in genetic changes. It's reversible, unlike genetic changes. It is reset each time in the germline. And I think it sort of presents a paradigm shift in the field of epigenetics. So this is non-Mendelian inheritance, which is important in mammals. So you don't only have genetic inheritance, but you also have epigenetic inheritance. It has an impact on development and disease, which I also told you about. These parental genomes are functionally different. Paternal genome affects the development of the placenta, and the maternal for the development of the fetus. They regulate growth and differentiation, brain and behavior. Also, they're important for stem cells and regeneration. And if things go wrong, they contribute to human diseases. And the important thing in the future is to find out more about the evolution of the imprinting. I told you that it is important for the development of placental viviparity, formation of the placenta and development in utero. And this might have contributed to adaptive radiation to give rise to diversity of mammals that we see now in the world. So I think this is a kind of a story of how you can start with a very simple question, and the answer you get can have implications beyond what you might imagine at the start. Thank you very much.